Good afternoon, everybody. Today I am going to talk about my research. I am a postdoctoral researcher at Department of Chemistry, University of Helsinki. I'm working in Kumpula campus. My work is about utilization of nanoplasmonic sensing in membrane drug interactions. So I will tell about those work that I'm doing at the moment. So before moving on to my real actual work, let me introduce myself. My name is Shishir Jaikishan. I have been living in Finland for last more than a decade. I'm from India. I have done my higher studies from University of Delhi. You can see on the map there that the arrow is showing yellow from pink India to Finland. How I got idea to come to Europe was really fascinating. I got selected for Nobel laureate meeting, Lindau, Germany. It was a very high profile meeting where I represented India in chemistry. And after that, I finished my master's in chemistry from University of Delhi. I applied for my PhD in biochemistry at Abo Academy under the supervision of Professor Peter Slotte, and got fortunate to do my PhD. After my PhD in Abo Academy, I continued there in the same lab for my postdoc. And after that, I went to very prestigious institute in the world, Weizmann Institute of Science, for my another postdoc. After that, I came back to Finland and started my postdoc at University of Helsinki this year, beginning January. So you can see that I have like versatile experience, and my field also kept on changing from every career jump. So it was from chemistry to biochemistry and biophysics of lipids to cell and molecular biology of lipids, and now I'm doing more of analytical chemistry, chemistry of lipids. So you can see that in all perspective, lipid is common in at every stage. So I will be talking a lot about lipids. Maybe many people don't like it, but it's a very good molecule that is keeping us healthy and functional in a healthy way. So this is the Nobel laureates that I was fortunate enough to meet at one place. It was a very good, excellent experience that I got a lot of motivation, and my learning curve started from there in an exponential way. So lipids, as you saw that I have been working on lipids for last many years at different stages, my PhD and postdocs. So lipids are very simple molecules which are organic and nonpolar. They are mainly composed of carbon, oxygen, and hydrogen, and definitely some other smaller percentage of composition of nitrogen and phosphorus, and some other also like sulfur and all. They are mostly glycerol and fatty acid. Glycerol and fatty acids works as their main building block of the cell membrane lipids. And some other common lipids are vegetable oils, which you are very much aware of, wax, then hormones, steroids, phospholipids and sphingolipids. I will be concentrating mostly on phospholipids and sphingolipids and sterols that I have been working on during my talk. So let's jump into that. Yeah, so this is a very simple picture. In this picture we have animals, we have plants, and then we have lots of human beings. So all of us, all living forms that we have on this earth are composed of proteins, lipids, nucleic acids, and they have their own role in maintaining the life in a proper way. If any balances is disturbed, then we get disease, and we have to get cured by eating medicines, and that's what we are doing at the moment. We are targeting mostly on lipids out of all the proteins and nucleic acids, and seeing their interaction with the drug, that how they are interacting and how we can be making it more efficient for therapeutic uses. And not just only big human life forms or plant or animals, any living beings, like viruses even, that you very well know of now. SARS-CoV, not a pleasant experience for all of us, but definitely they are there. They have also lipids as their membrane. Animal cells, plant cells, or bacterial cells, everyone is having the same compositions for their functioning, lipids and proteins and nucleic acids. So I will slowly go into the mode where we will go from higher life form to our molecule of interest, lipids, and then from there onwards, like what I'm doing exactly with lipids. So that was a 3D cell model that in the previous slide you saw. Now it is a 2D model. Here you see that in a normal cell we have nucleus, we have mitochondria, we have vacuole, centrosome, a cell wall, depending upon the kind of microorganism or cell type we talk. And out of all those things, we are, or I am here trying to focus mainly on cell plasma membrane. Cell plasma membrane is a kind of like enclosure of a cell. It provides the security. It provides the structural intactness, which contains everything inside it, and let it function in a best possible way so that the cells survive. But recently it has been found for last maybe 30-40 years that these cell membranes are not just for structure, but they play a very important role in normal functioning of cell also via viral entry or via signaling also. So now you see that I have marked cell membrane in a red box. So if we zoom it, then it is looking like that, where we have like lots of proteins embedded into the cell membrane, and the whole C is made up of lipids. So these lipids or phospholipids, mostly when we are discussing about the cell membrane, they contain all these functional proteins also, which are further supported by lots of complex structures. So for simplicity, I am trying to make it more simple so that we can get to the gist of this slide or presentation. From that, I have made another remark which shows just the lipid part of whole cell membrane, where we have lipid molecules, phospholipids, having two forms, a nonpolar part and then the polar part. Polar is mostly exposed, where we have more aqueous exposure, aqueous environment, and then we have nonpolar part, which is hydrophobic, and it kinds of like not allowing all the hydrophilic molecule very freely into the cell. It's very selective. Yeah, so it's a continuation from the same picture. You see that the lipids are having a hydrophobic head and then hydrophobic tail, and if we zoom it, it something looks like that. That we have water molecules jumping or touching the head part of the phospholipid, and the hydrophobic part is embedded inside the hydrophilic head. So now I will go into more deep of the structure of these phospholipids, like what kind of structure they have in chemistry, and how is the variation, and what are the possibilities of different lipid forms that we have in any cell. Yeah, so this is very simple representation of thousands and thousands of lipids that could have been present in our cell, irrespective of plant cell, animal cell, bacterial cell, or viral cell. We have, as I have told you in previous slide, that phospholipids are mostly having a backbone of glycerol. Attached to it, we have at two positions, SN1 and SN2. The orange part that you see, fatty acids. These fatty acids could be saturated, this could be branched, this could be unsaturated. Mostly we have unsaturated, but when it requires a saturated one, it can synthesize on spot also, or it can have like localized saturated fatty acids also with the phospholipids. Regarding head group, which is hydrophilic, we can have like different of options in phospholipid, like it could be choline, which is mostly very common in our cell. It could be from 50 to 80, 90% depending on the cell and then the kind of environment they are living. Other head groups are also very prominent in phospholipids. It could be ethylonamine, it could be serine, it could be inositol, it could be glycerol also, even though it's kind of like backbone which is present in the interface, which is like green part and the orange part there. So it can be present as head group also in the head region, in hydrophilic region. And definitely they have different charges depending upon the pH also, or the cell environment present in our body. Apart from phospholipids, we have sphingolipids also, which is almost similar in structure but have the backbone a bit different. You can see that serine is present there on the right side at the backbone instead of glycerol. And the head group could be choline, or it could be a number of different kind of head group present with the complex sphingolipids. Ganglioside are typical examples which are really very important in kind of signaling and functioning of sphingolipids. Sorry. So how these phospholipids are formed, how they are like covering the cell structure that we saw earlier. It's a very entropy driven process. So whenever we put these kind of phospholipids in aqueous media or water, due to the unfavorable interactions of their hydrophobic part with the water, they tend to protect their hydrophobic part and expose their hydrophilic part towards the water side. So when we put these lipids in a dry film format into the water, it immediately forms multilamellar vesicles. So multilamellar vesicles are like we have a circle inside circle inside circle. It could be like tens, it could be like 2, it could be like just 1 also, as you can see. And the sizes can be from few micrometers to few tens of nanometer, depending upon the temperature, composition, and all these kind of factors of or size also of the phospholipid. But if we want to have a uniform liposome or vesicles that we call all these particles, we have to treat them through severe physical process. It could be probe sonication, it could be bath sonication, it could be extrusion, it can be freeze thaw. I don't want to go into details of all those process method, but it's very simple. It's not very complex. And when we go through all these processes, then it can be like a uniform in dispersion, uniform in sizes, and then we can do more research, or you can apply for it in a different way. So here you see that this green part, we have space inside, which is mostly aqueous, and then we have blue part there, which is hydrophobic, and those are 3 to 4 nanometers in size. Yeah, when we are talking about self-aggregation, as you can see in the previous slide, that it forms different kind of multilamellar vesicles. So it's necessarily not possible to have these kind of spherical structures only. It could be like many different types of morphology that they can exhibit depending upon the temperature, molecular shape of the lipids, and the condition of the lipid water mixture, like ionic strength or concentration of the buffer. So you see that we have like lots of possibilities of these lipid molecules. It could be micelle if it has a bigger head group and a smaller tail. It could be a reverse micelle if the structure is in a reverse way. It could form prolate micelles or hexagonal phase. In the center we have large multilamellar vesicles, as the same in the previous slide you have seen. And they can form bicells also if we have two different compositions, having like one with longer tail and smaller head group, and then we have head group with very small tail. So those kind of structures tends to form bicells. And every single composition and structure has some therapeutic or kind of functional use that people tend to harness while manufacturing these vesicles or morphological structures of lipids. So use of liposomes. Liposomes are used for a number of purpose, a number of therapeutic use. I'm not like focusing every single of them because it will take too much of a time. But as we have discussed so far, that mostly these liposomes have a core which is of aqueous nature, and then we have on the surface where you can have like again aqueous environment, but in between of that bilayer you have a hydrophobic environment where you can store hydrophobic molecules, and inside you can store aqueous or hydrophilic molecules. You can reconstitute proteins or different forms of structures, or for example esterol, DNA moiety, to have it like targeted to the site where you want to deliver all these drugs present inside the bilayer membrane or inside of the liposome. So you can have like lots and lots of functionalized forms of liposome to have like different functions. I will show in the next slide like what kind of molecules, drugs that we are encapsulating in these liposomes so that we can deliver those drugs. Those drugs are mostly very insoluble in all kind of media, so it's very difficult to deliver to any patient. So we try to inculcate all those drugs into these kind of structures, liposomes, which tends to increase the absorption rate of those drugs in our body. For example, I have put here a slide where we have few of the drugs like cephalexin, levofloxacin, which by itself are kind of like not good, very well absorbed in the body, but when we inculcate all those drugs in lipid liposomes of different compositions, we prepare with different methods, we can deliver these drugs in a best possible way to any patient. So this way we go down in amount of drug also, because the absorption rate is increased with this kind of delivery mechanism, and it's more effective. So we are kind of saving drugs also, and those drugs are very expensive also and very difficult to make also. So definitely these liposomes are a very good carrier, and you might have seen during the corona time that this Moderna company was having these RNAs encapsulated in between these liposomes and then delivering it to the person. So that's very new field at the moment regarding use of liposomes for gene delivery and vaccines. So this was all about, for example, lipids. Maybe it's too much to digest with such a short time, but it's a very simple thing. When you put a bit of more effort, you will immediately get to understand that how and what these lipid molecules are. So this is the half part of my research. Other half part will be the technology that I'm using exclusively in the lab. We are using lots of different technologies like capillary electrophoresis, then asymmetric field flow fractionation, and DLS, dynamic light scattering. And I have been used many other systems techniques also, but I will focus mostly on nanoplasmonic sensing for now. This technique is very unique and very recently developed, and it's still being kind of like in nascent stage. It's not being like in full-fledged mode. So we are one of the few labs in the world which is using this technique with for lipid research. This is very simple method where we have been always using this with a different form of application. So generally it was surface plasmon resonance. Here we are doing localized surface plasmon resonance. So for SPR we have the resolution of say few hundreds of nanometer. Here we go down to a few tenths of nanometer. It's a very simple technique based on refractive index of the system. So it's something like that. You have a sensor, and then when you have any different molecule attached to it, it will report some kind of refractive index. When you put another molecule on top of it, for example, here we have Bayer chip, it has its own refractive index. Then we put a PEG molecule on it, and then antibody, and then exosome. So every single system has a different refractive index, and that is being reported in the in the detector, obviously. Yeah. So here you see, so when we put light on gold surface, not like continuously, but with kind of like, here and there spotted or engraved. So it shows plasmons. Plasmons are electron-rich cloud, which has its own properties. And when we put anything different on it, it changes the refractive index of the system, and that's what we are detecting in the system. And those gold discs are of 200 nanometer or 100 nanometer with a separation of few tens of nanometer to 200 nanometer, and height could be like 10 to few again tens of nanometer. And they are coated with silicon dioxide, silicon nitride, or titanium oxide, so that the longevity of the gold surface is bit increased. So we can do a lot of research or a lot of biosamples, study of biosamples on these sensors. It could be lipids, proteins, viruses, biominerals, hydrogels. This is the instrument. This is one of its first kind in the world that we are using at the moment. It's a very simple instrument, but very sensitive for our setup. These are the sensors that we use. So inside the circle we have gold nanoparticles encoded on the silica surface or glass, and it could be of many different sizes. So you can have nano hole, grating, nano arrows, nano rods, nano islands. These are very highly zoomed pictures under maybe electron microscope or scanning electron microscope. So what we do is we put liposomes, those kind of structures that you saw in the previous slides, on those sensors, and then we do various kind of interaction studies. So we put liposomes on them and get it absorbed with many preconditioning features, and we then get graphs on the screen. Depending upon the nature of the sensor, we can have a graph which is like that, or which you can see on the top, or it could be like a sigmoidal, which is on the lower part of the graph. So in lower part we have all these intact vesicles coming to the contact of sensor, and then it bursts, and then it lies on top of it like a supported bilayer. But in case of titanium oxide, which is like in the top of the graph that you are seeing, it will remain intact and be like that. So we can do interaction studies in both kind of setups or systems. So here it is the same thing, and we are putting all those lipids via flow mechanism, and the flow is always continuously being allowed, so that we have drugs also injected on top of these vesicles, and see their interaction, how the refractive index is changing. So here we have tried to do the transition temperature studies. So it's not like any kind of interactions, but just the transition of liposomes that we are seeing with the increase in temperature. Yeah, so at gel phase we have like intact circular vesicle, spherical vesicles, but as we increase the temperature it gets deformed, and these deformations are kind of measured or noted by detector, and then we do some kind of mathematical calculations or so that to get the changes in the influx point, and that points are close to the pre-transitions or the transitions of the liposomes. Yeah, so this is a bit complex graph. I would like to skip it. And that's how we have found the transition temperature of these lipids, all these phosphodiecholine molecules. As we increase the chain length, we saw the increase in the transition temperature. So here we have tried to see the interactions of these ionic liquids with our liposome system. So we have three kinds of ionic liquids that we have selected here. So these are organic molecules which are utilized for synthesis, dissolution of cellulose, lubricants, electrolytes. So when we expose these liposomes to these ionic liquids, they get disrupted or kind of like deformed. Yeah, it's the same thing, same slide, that the structure that we saw. In the first graph, we will see intact vesicles. In the second graph, we will see supported bilayer. And then we try to have the interactions of these ionic liquids with these kind of liposomes. So here we see that when we put ionic liquids on these POPC liposomes, it doesn't show any kind of interactions. So that means it didn't show any deflection in the baseline. So that's why we say that there is no interactions with this ionic liquid with POPC liposomes. But here you see when we applied P1444 OAC ionic liquid, it completely deteriorated the liposomes. As we put water after the ionic liquid exposure, you see that the curve goes literally down. That means the whole bilayer is being destroyed. It's the same. We have increased the concentration from 0.15 millimolar to 1 millimolar. You saw the effect that it's kind of getting completely destroyed on the sensor, the liposomes. It's same. Another example. So here we have included cholesterol. So when we change the composition of liposomes, so obviously these kind of structures can also show different properties. So it's very important to see the composition of lipids also, to see how they are behaving on the sensor, and definitely after immobilization, how they are interacting with the drug molecule or any molecule of interest. So it's not just about liposomes. So if we change the sensor composition or structures like nano island, nano rods, we can select a higher range of molecules also. As for example, we have 10 to 20 nanometers. We can go higher in 100 nanometers of resolution. So this technique is very robust and flexible also for different kind of studies. So recently people are more interested in exosome studies also to utilize this technique. It's same here. We have different kind of antibody interactions with this technique. So comparing all this technique, we see that nanoplasmonic has a lot of edge when it comes to simplicity, sensitivity, throughput, and translational potential as compared to its conventional SPR or SIRS and fluorescence, which is equally good and being utilized very nicely throughout the research community. But definitely nanoplasmonic sensing is putting up more benefits for this kind of research. So in conclusion, it is a very simple instrument, but very sensitive also, which has the resolution of few tens of nanometer, and we can do lots of interaction studies. It could be analyte, it could be drugs. I purposefully didn't put any drug, but I put drug instead of drug, ionic liquid, so that our research is kind of like bit saved. It's a very new research that we don't want to expose right now. And we can do both kind of intact liposomes and supported bilayer studies, because those have different properties and different way of showing the interactions. So it would be really nice to see any kind of drug interactions with lipid in both kind of systems, intact liposomes and supported bilayers. I hope it was easy to understand. Maybe not too easy, but I try to make it like in a simplest possible way. I hope you will have some questions to answer.